Hello, and thanks for joining us for today's webinar with Inoviv. My name is Karen Davis, and I'll be the moderator today. In this webinar, Inoviv's Senior Vice President, Ben Gonzales, and Chief Scientific Officer, Ernestas Sirka, will discuss NeuroKey-3, a fully quantitative multiplexed IP-MS assay that measures key low-level markers linked to neurodegeneration. Senior Vice President Ben Gonzales is a veteran in the field of proteomics with over 2 decades of leadership experience in the biotechnology and pharmaceutical industries. He has an impressive track record of successfully developing and commercializing innovative technologies for diagnostics and research and development applications. Chief Scientific Officer Ernestas Sirka is a biochemistry expert with extensive experience in clinical test development, distinguished by roles at Pfizer and University College London. Notably, he has pioneered the development of a unique targeted proteomics platform and multiple patent-pending biomarker tests for drug development applications across several therapeutic areas. So, without further delay, I'll turn the time over to Ben and Ernestas.
As mentioned, I'm Ben Gonzales. I'm the Senior Vice President of Commercial here at Inoviv. As a brief bit of background, my travels have taken me from the likes of Bruker to SomaLogic, to General Manager at Biognosys, and now here at Inoviv. Having been in the omics space for some time now, I've seen that so much of proteomics has been focused on discovery that it really seems like the functionalization of proteomics is almost an afterthought. It's one thing to ID a new set of biomarkers and yet another to see them used in development. And this is what we'll be talking about today. I'm also happy to connect. On your screen, you'll see a QR code which links to my LinkedIn profile if you'd like to connect. So let's begin with a quick chat about biomarkers. Well, I'm sure you all know I'll highlight that a biomarker is really any monitorable marker that can help us understand what's happening biologically. This could be as simple as heart rate or blood pressure, and for our purposes here, we're exploring much more deeply at the molecular level. While some classes of biomarkers are well understood and characterized, and even used routinely in the medical field, like genomic markers, the evaluation of protein biomarkers presents a much broader challenge. At this time, I think most biologists recognize the importance of measuring proteins as a marker of disease or treatment, but proteins are tricky. They cover quite the dynamic range, and equally challenging, they come in a lot of different shapes and sizes, literally, even when you consider a single protein. For example, tau, a well-known neurological protein marker, has different phosphorylation sites, known as post-translational modifications. It aggregates, and it is known to have isoforms and truncations. When we consider this, it's important to have a tool that allows for rapid and confident ID of biomarkers that also provides accurate quantitative measurement of said markers and is really able to distinguish between all the possible flavors, including any post-translational modifications. And here at Inoviv, this is where we shine. We develop multiplex protein biomarker assays that can achieve unparalleled sensitivity, as you'll see here today with NeuroKey-3. Our technology supports the rapid assessment and monitoring of protein biomarkers through pre-validated assays. No other company is able to achieve the level of sensitivity that we can, combined with industry-leading reproducibility in an off-the-shelf assay that's available to support researchers without having to concern themselves with any of their own development. Our technology combines the best of both worlds, using affinity modalities where needed to support as low as picogram sensitivity while offering high-throughput multiplexed workflows using the gold standard technology of mass spectrometry. Mass spec also provides extremely high confidence in identifications and quantification and the ability to see multiple epitopes to any given protein while avoiding the pitfalls of technologies that only look at 1 or 2 epitopes. Working with Inoviv, we can support you with a service model. We provide targeted, validated, fully quantitative multiplex proteomics assays with no prior knowledge required. Really as simple as 1, 2, 3. We provide consultation for our clients to determine the ideal solution from one of our pre-validated or bespoke assays. We intake samples and analyze them via mass spectrometry. And finally, we deliver a report to our client with fully quantitative output to support their clinical trial workflows. On that note, it's worth highlighting that we can do routine custom development through our Inoviv platform as well. which allows for full development from discovery to validated quantitative results, even if you don't know where to start. And with that, let's get to why you're really here. I'm really excited to introduce you to Ernestas Sirka, our CSO here at Inoviv. He's a seasoned biochemistry expert coming from roles at Pfizer and UCL, earning recognition for his work in journals like Nature Medicine and The Lancet. At Inoviv, he has pioneered our targeted, high-sensitivity multiplex proteomics platform and patent-pending biomarker tests for drug development. On a personal note, I can say that Ernestas has probably forgotten more about proteomics than I'll ever learn, and I'm happy to share the floor with him today and for him to take us on a journey on how we validate quantitative mass spec proteomics assays and how this has been applied to NeuroKey3. Take it away, Ernestas.
Great. Thanks, Ben. I'm happy to take over from here. And I'll be just a second whilst I boot up my slides here. So following on from what Ben was talking about, I also wanted to cover a few things today. And I'll add a few bits of context on the biomarkers in clinical trials. Then I'll highlight one of the biomarker tests that we've built recently. And I'll show you the results we got from its analytical validation. And I'll also touch on the clinical cohort data. And there's going to be time for questions at the end as well. So it's probably not a secret to this audience that drug development is challenging. A lot of drugs in development don't make it. And there are multiple reasons behind it, but one aspect can be data reliability. And here I mean biomarker data. On top of that, regulators such as FDA also have standards for data reliability in a form of bioanalytical method validation guidelines that are quite stringent. And at the same time, the regulators are encouraging biomarker strategies. Now, if the data is not reproducible or it's highly variable, if you're not quantifying the target you want to quantify, or if you can't do it in the first place because the sensitivity is lacking, that could mean that things go slow from phase 1 to phase 2 and beyond. And in the worst case scenario, trials can fail as well. But there are solutions for this. And over the next 20 minutes or so, I'll be highlighting how we're addressing these challenges, where we are simultaneously achieving high reproducibility and precision alongside high specificity and sensitivity in order to accelerate drug development and increase the chances of success. And one of these solutions is immunoprecipitation combined with mass spectrometry, or IP-MS for short. It uses antibodies to enrich the analyte of interest to boost the sensitivity, whilst maintaining the specificity and reproducibility which comes from mass spec-based detection. IP-MS is currently gold standard for quantification of low-level biomarkers across therapeutic areas, and that's certainly true for neurodegeneration and Alzheimer's disease as well, which is our focus today. For those of you who don't usually work on Alzheimer's disease, it's the most common cause of dementia. But fortunately, we now have the first few disease-modifying therapies, such as lecanemab or donanemab, that slow disease progression. There is, however, the need to diagnose patients in a timely manner, timely and also scalable manner, so that they can benefit from these new therapies. And there's also still a need to stage the disease, predict its course, and keep assessing the efficacy of other drugs in development and accelerate them through the development pipeline. Those new drugs could be even more effective. They will be addressing other aspects of this multifactorial disease, in addition to removal amyloid, which is what the first disease-modifying therapies do. And they could also come with easier routes of administration as well. But we'll absolutely need to know if these new drugs are engaging their targets early on in their development. And we'll also need to understand whether these drugs work or not at a molecular level. And if so, through which pathways. Very likely, we'll need to give different drugs different patients at different stages of disease once these new drugs are approved. So we'll really need to untangle and characterize all of this complexity at the molecular level if we want to have effective treatments. And that's where biomarkers come in. So recently, the National Institute on Aging and the Alzheimer's Association in the US have published criteria for diagnosis and staging of Alzheimer's disease. These guidelines are intended to inform not only clinical care, but also research, which includes new drug development. I'm not going to go into the detail here on this table, but at a high level, multiple biomarkers are now recommended to address various aspects of Alzheimer's disease. And we at Inoviv have seen this trend towards multiplexing accelerating over the years. Also, FDA has started approving drugs based on biomarker data in other neurodegenerative conditions, such as tofersen in ALS. And that is why we have recently developed a multiplexed IP-MS biomarker test. We're calling it NeuroKey, as it covers key aspects of Alzheimer's disease pathology, from inflammation and synaptic degeneration, all the way to core Alzheimer's disease biomarkers. But whenever we build a biomarker test, regardless of the disease process or the therapeutic area, we want to make sure that the analytical performance of these biomarker tests is as robust as it can possibly be, so that we can rely on the data that biomarker test is going to produce once applied to a preclinical or a clinical sample set. And that means validation. Now, validation is a fairly lengthy topic with multiple scenarios and dependencies, but I'll highlight a couple of things here. So here is a non-exhaustive list of the analytical validation parameters we characterize for our targeted and multiplexed biomarker assays, where we are measuring biomarkers in clinical trials or preclinical studies. I'll be highlighting a few of these parameters as we go through this section, but you may still be wondering, why do we do this validation? Why does it matter? And we do this because we need to know for ourselves how good our measurements are and if we can rely on them. And also our clients want to know the same thing. On top of that, as I mentioned, FDA has standards for data quality in a form of guidelines for analytical method validation. And this is becoming more and more important because, as I said, FDA has started approving drugs based on biomarker data alone in some circumstances. And if we zoom out, overall, more and more emphasis is placed on biomarker data during drug development in the field as a whole. Biomarkers are seen as insurance policy on clinical trials. So we really need to know how good the data is. And over the years, we've developed a method for biomarker test development and also successful validation as well. And it largely hinges on three things. The first one is automation. So we use robotic liquid handling systems, usually operating in a 96 well plate format. And we also use proprietary workflows where we essentially optimize pretty much every single variable in the whole multiplex IP-MS process. From how much sample to take, to what to dilute it in, to how to digest the proteins once they're extracted from the sample, not to mention which antibodies to use, or should we even use antibodies? There are other immunoaffinity reagents that might be better suited. And finally, internal standards. These are super important as well. And one of the critical bits there is to add them at the very start of the sample preparation process so that they can compensate for any variability during the quantification process for every analyte in a biomarker test. And these are the results that we're getting. So on the right here, it's neurogranin. it looks like a very neat single straight line, but actually there's 15 measurements behind this calibration line effectively. We had multiple quality control samples here, spiked and unspiked, surrogate matrix, as well as real life plasma samples as well. And we've done this 15 times, multiple times on the same day and multiple times on different days as well. And no matter how many times we do this, we're getting the same result. And visually, you can see here really tight error bars, or basically no error bars at all. That's how tight they are. So what that tells us is that the assay is sensitive, it's accurate, and it's reproducible. And here on the left, we have a numerical representation of this validation. So as I mentioned, we follow the FDA biological method validation guidelines. And just to highlight a couple of things here, if we look at precision, again, in surrogate matrix, as well as in real life clinical samples as well, we're getting single digit CVs. And that tells us that the assay is precise in surrogate matrix, as well as in real life plasma samples as well. Now, if we look at interleukin 18, which we're also measuring in the same multiplex IP-MS assay, it's the same set of excellent results. You can see that visually on the graph on the right. And again, numerically, if we focus on CVs again, perhaps even more impressive intra and inter-day CVs, both in surrogate matrix as well as in real live plasma samples as well. Now, we also build multiplexed non-IP-MS biomarker tests, such as our broad Alzheimer's disease plasma biomarker panel, which covers multiple biological processes at the same time. Here I've got one of those markers out of that broad biomarker test, adiponectin as an example. And once again, if we look on the right to the right at the graph, we're seeing the same set of excellent results. It's really the same story for the third time in a row. So what this shows is that our biomarker tests are accurate and precise across the full range of protein concentrations or abundance level, if you like. And in fact, we've recently broken precision records. So we got a 2% CV in real life clinical samples, which is quite rare to achieve this kind of performance in a clinical sample in the actual matrix. So what we're showing here really is that we're routinely achieving precise and reproducible measurements across all of our targeted proteomics platforms. And you may be wondering why this matters. Why do we care about this? And it matters because this kind of performance essentially takes away the technical variability. So it's easier to show pharmacological variability. It's easier to show a larger effect size, or we're actually maximizing the chances of showing any kind of difference on clinical or preclinical study in the first place. And at the same time, the data is fit to submit to regulators. Now, for the final section, I wanted to touch on our initial data in the first public clinical cohort, where we applied this multiplexed IP-MS biomarker test, NeuroKey 3. So let's start with neurogranin, where we found that neurogranin was decreased in Alzheimer's disease plasma compared to age and sex matched controls. You can see that in the graphs here on the right. And we've been able to confirm this trend by looking at multiple surrogate peptides, or in other words, different parts of neurogranin protein. Up next is interleukin 18, which we're also measuring as part of the same multiplex biomarker test. Interleukins are usually low level and therefore hard to measure reliably in plasma. But in our data, we were able to quantify this successfully and have detected a statistically significant difference between different patient groups. Again, you can see this here on the right. So here, plasma interleukin 18 levels were lower in patients with Alzheimer's disease compared to age and sex matched controls. And finally, we saw the same result with BDNF as well. And what we're doing next here is we're applying the same multiplexed neurokey biomarker test to larger public cohorts of Alzheimer's disease with matched CSF samples where available. And we're doing that because the literature is scarce and inconsistent on these biomarkers in plasma in Alzheimer's disease, particularly where the data is generated with validated assays. And so now, now that we know that analytical performance of the assay is rock solid, and that we're tracking multiple proteins and multiple parts of these proteins at the same time in a single assay, we're really in a strong position to apply this to further public cohorts to advance the knowledge in the field here for the benefit of the whole society and community who's working on Alzheimer's disease and trying to develop treatments for the disease as well. And at the same time, we're already working with drug developers, applying this assay to large clinical trial cohorts privately, seeing some exciting results post-drug treatment, and we'll be sharing those data sets publicly as well once our clients make them public too. And I'll finish with this lovely quote from one of our clients, T3D Therapeutics, who is a clinical stage Alzheimer's disease company. And what they said here is that applying Inoviv's biomarker analysis systems in their phase 2 trial has enabled a comprehensive assessment of drug efficacy. And also, the high quality data generated has informed the design of future clinical trials as well. So those were all of the data sets we wanted to show you today. And we covered quite a bit of ground. So thanks for staying with me. Thanks for attending in the first place. And I think we still have time for a few questions.
Yes. Thank you, Ernestas and Ben, for a great presentation. So we do have some questions, and if more arise, a reminder, you can submit them using the Q&A tab. So let's dive in. First question is this applicable for other matrices, specifically CSF?
Absolutely, yes. Although we focused on plasma, it's absolutely applicable for CSF as well. CSF is actually even a lot easier to work with compared to plasma. So CSF will work, and if we zoom out, generally, our technology platforms are matrix agnostic, so this will absolutely work for other matrices as well.
Okay, and can you clarify your multiplex IP-MS process?
Yes, it's not an easy one, and there is quite a bit of process there. So at a high level, it's basically a lot of optimization. So as I mentioned, we've optimized pretty much every single variable there is. So how much plasma to take to begin with, what to dilute it in, how to digest the protein so that they actually fit into LC-MS system once they're extracted from the plasma. And as I mentioned, what do we even use for enrichment? Which antibodies do we even use? Antibodies? Do we use something else? So there was quite a lot of optimization just on the sample preparation side of things. And then we also needed to make sure that all of that works in an automated way as well on automated liquid handling systems. So it's in short, it's a long process where we optimize basically every single variable along the way.
How does GFAP IP-MS sensitivity compare with other assays like Samba?
Good question. I don't think we have direct comparative data. I can come back to you on that, but we know that we can detect a GFAP in biofluid samples. And I think we can follow up directly with a data set. I'll just need to double check what exactly we can share, but I know that we have enough sensitivity to detect it in biofluid samples.
And would this work for dried blood spots?
With this work for dried blood spots, let me reflect for a second there. For dried blood spots, IP-MS, probably not. We probably need more volume, more material there. But what would work for dried blood spots is, we could focus on different set of analytes. So if someone has, a specific set of analytes in mind, there are certainly some molecules that would work for dried blood spots. It's a very limited sample volume there, and definitely not everything works in dry blood spots, but there are analytes, for instance, lipids work beautifully in dry blood spots. Some of the proteins work beautifully in dry blood spots as well. So it'd be great to dig into the specifics there. Again, hopefully we'll have a chance to follow up directly to get a bit more detail around the context there.
How do we work with Inoviv on a study?
Ben, do you want to take that one?
I can probably take that one. So we do this via a consultative basis that moves into design. So if somebody is interested, for example, on that GFAP question that had come up, in either creating a bespoke assay or using one of our pre-validated assays, we would set them up with one of our business development folks. We would bring in one of our technical specialists in order to make sure that the study is well designed and well powered and really consider what the outcomes are. Oftentimes in our experience, a lot of folks are focusing on moving into phase 2 and phase 3 clinical trials. So we would understand the regulatory requirements, GLP and CLIA and so on from there, and then be able to provide a proposal and then, of course, run and deliver data based on that.
Next question. The MS variability is impressive, but less than what's commonly seen for MS. Can you explain?
Less. Yeah, I think if I understand the question correctly, I think it's, I think other mass spectrometry approaches could have even, higher CVs. And what we've done here, as I mentioned, we really optimized basically everything there is to optimize in the whole process when building these IP-MS and actually non-IP-MS assays as well, and we are getting really excellent performance. So I think it just really comes down to just really how much optimization we've done here to make sure that the platforms are robust, and then we can build all sorts of different biomarker tests on top of those.
Can we customize NK3 for our own use?
Ben, do you want to take that one?
Yeah, I can probably take that, and then maybe I'll kick it back to you, Ernestas. So we can certainly develop bespoke assays. And so whether it's a combination of the markers that you've seen here today, and maybe even overlaying what we call our MFPs, my favorite proteins, to go along with it, that's something that we could likely do under our InnoKey workflow. And so that's where anything from a discovery approach all the way through to creating that validated quantitative proteomics panel. But Ernestas, I don't know if there's anything else you wanted to add to that or not.
I think you covered it.
Right.
Awesome. Well, if any other questions arise or you would like additional information, please visit inoviv.com, or you can reach us directly at info@inoviv.com. Thank you again, Ben, Ernestas, and the Inoviv team, and we thank you all for attending today's webinar. Have a great day. Thanks, everyone.